So for our next topic, we're going to talk about the link between DNA and the nucleus and actual visible traits we see in the world. We're going to start with something called transcription. Okay, so transcription is the first step of something called the central dogma. The central dogma is just a statement, a theory, that states that there's a link between DNA in the nucleus of your cell and proteins, which are the things that we actually see in the world. So we've talked about some proteins like enzymes that'll do cellular processes, but proteins can also be things like pigments or muscle fibers. Proteins do a lot of different things that show up as visible traits. Okay, so the central dogma states that there's a link where DNA provides the blueprint for proteins using RNA basically as an intermediate step. So DNA makes copies of itself that get into every cell by replication, which we've discussed already. But in order to make a protein, DNA first needs to be transcribed into RNA, which will then be translated into a protein. So transcription is the first part of a process going from DNA up here to RNA. This is our transcription part. And then from the RNA, we're going to take that and use that as code to make a polypeptide. And polypeptide is just an unfolded part of a protein. So this is a big roadmap for where we're going to understand this whole process. Right now, we're up here at this DNA to RNA part. Okay, so when we're doing transcription, you need to remember the word transcribe means like to write something down. So what we're doing is taking DNA, using that as our blueprint, and we're writing it down in this language of RNA. And specifically, what we're going to be making is something called mRNA. The M here stands for messenger. We'll see later that mRNA is able to leave the nucleus and take that as a message to other places in the cell. Okay, so we take our DNA and we're going to read each gene individually. So we're looking here at a gene. So this whole big long pink thing is our strand of DNA in a chromosome. This chunk here is a specific gene. So genes have a region that codes for our protein. This is the central part, this transcribed region. So this is here, this pink part that we've zoomed in on and put in brackets. There are also parts of the gene that regulate how and when that protein is made. We're going to have a bit at the front here in blue called the promoter. So the promoter is going to signal basically that this is the start of the gene. We're going to have a terminator at the end, signaling that this is the end of the gene. Okay. There are also potentially additional regulatory sequences that are the site for binding things that tell you to make this faster, make this slower, something like that. Okay, so we have our gene on DNA. We're going to use that to make our mRNA transcript. Okay. So the first step of this is called initiation. So initiate, just beginning, right? And in initiation, we're going to have our RNA polymerase bind to this blue part, the beginning of the gene, the promoter region. Okay. So here in purple, we see our RNA polymerase. It's got some sort of helper molecules. Here you can see one called the sigma factor, helps it recognize the promoter region. But RNA polymerase is the important thing to remember here. So RNA polymerase is kind of like DNA polymerase. When we talked about DNA replication, we had DNA polymerase pulling in our new DNA nucleotides. RNA polymerase is going to pull in some RNA nucleotides in order to help us make a copy of the DNA that's written in this new language of RNA. Okay, so our RNA polymerase is binding into the promoter region, and the way that the RNA polymerase is shaped, it basically has like a little prong that's able to kind of pull apart the DNA and create a little bubble where RNA nucleotides will be able to get to the DNA and will be able to match up. When they actually do the matching up, that's our second step, elongation. So we have our sort of little pronged RNA polymerase. It's moving down the DNA, keeping this little bubble open, and it's going to transcribe the region of the gene that gets transcribed, that central region. And what it's doing as it does that is it's pulling in RNA nucleotides and linking them together. Now the RNA nucleotides match up to our DNA template, and they pair the way that DNA pairs to itself. So if there's a G on your DNA template, there will be a C on your RNA. If there's a T on your DNA, there will be an A on your RNA. But the difference is that RNA doesn't have thymine, that letter T. So an A on DNA is not going to give you a T on RNA, it's going to give you a U. U is for uracil. It's a special nucleotide that only RNA has. So if you ever see a sequence of letters and there are U's in it, we're looking at an RNA. Okay. Another thing to note is that as RNA polymerase is doing this transcription, it's actually only reading one of the strands of DNA. The other one it ignores. Our mRNA is going to be just a single strand, unlike DNA, which is a double strand. Okay, so our RNA polymerase is going to get to the end of the gene. It's going to hit what we call the terminator region. So it's going to detach from DNA. When it detaches from DNA, that little bubble in the DNA closes, and the mRNA transcript that it's been making falls off. Everything falls apart, so we now have this mRNA transcript free in the nucleus. So remember, DNA is stored in the nucleus. It's always in the nucleus. So this whole process is happening inside the nucleus. Okay, so there are some additional processing steps to get us to mature mRNA. We're not going to talk about them in detail, but in case you hear about them in the future, it's true that some parts of this transcript, so some parts essentially of the information in the gene, get cut out. They don't end up in the final copy of this RNA. Those parts that get cut out happen to be called introns. We also add sort of like caps and tails, so things on the ends that make it easier for this molecule to interact with the rest of your cell. Okay, so this whole process of transcription is taking place in the nucleus. So here we see our cell. Here's our nucleus. Our DNA is always stuck in the nucleus. It's stuck inside this membrane, which protects it and keeps it from leaving. So we do transcription, write down our mRNA transcript, which is like a copy of DNA, but it's in RNA. And RNA, our mRNA, is actually able to get out of the nucleus. So it's going to leave like through one of these nuclear pores or something, these little doors in the nucleus. And our next step is going to happen out in the cytoplasm. It's actually going to happen using our ribosomes. So we'll talk about that next.